The oocyte still arrested in metaphase 2 will complete meiosis II after sperm entry. This will result in the formation of a mature ovum and a second polar body. The ovum's nucleus will then swell, forming the AFAB, or the female pronucleus, and it now will await its AMAB counterpart to complete the union of gametes. And that's what we're seeing here. So let's walk through the final 4 steps of fertilization. Step 1, the surge in intracellular calcium will trigger the oocyte to complete meiosis II. This will yield the ovum and a second polar body. The ovum's nucleus swells to become what we call the female, or AFAB, pronucleus, which sets the stage for genome fusion. Next, the sperm's nucleus will also transform. It enlarges and becomes the male, or AMAB, pronucleus. This migration of pronuclei toward one another is choreographed by the cell's cytoskeleton, preparing for a coordinated union. As the 2 pronuclei move closer, their DNA replicates. In this, a mitotic spindle will begin to form between them. Their nuclear envelopes will disintegrate, allowing the maternal and paternal chromosomes to mingle. This sets the stage for the first mitotic division of new human life. And lastly, the chromosomes align on the spindle and unite. This moment marks true fertilization, when maternal and paternal genetic material combine into a single diploid genome. The result from this is a zygote, which is the first cell of a new individual with its own unique genetic identity. Now we'll pivot from conception to starting the process of development. And the zygote is the ultimate stem cell. It's what we call totipotent stem cell, which is the most versatile type. From this single cell arises every single tissue and organ in the human body. This remarkable ability and potential is orchestrated through the processes of cell differentiation. This process by which essentially cells become specialized by selectively expressing parts of their DNA, not all of their DNA. So let's pause and think about some review questions. These are some review questions that you should be able to answer throughout this lecture. And so, how do cells become so different despite having identical genomes? Think about the role of proteins, gene expression, and epigenetic regulation. Also consider how stem cells vary in potential from totipotent to unipotent, and what that means in both embryology and in regenerative medicine. Every cell in your body contains the same DNA. I think that's important to first understand. Yet a neuron is a profoundly different cell, as we can see here, than a muscle cell. And this is because of the gene expression component. Be it via selective or through selective activation and silencing of genes, cells will take on a unique identity. This concept is foundational, not just for development, but also in understanding things like cancer, stem cell therapy, and gene editing. So the zygote, as I mentioned, is the ultimate example of cellular potential because it's totipotent, meaning it can give rise to all the cells of the body and the extra embryonic tissues, like the placenta, for instance. As development will progress, cells become increasingly restricted in their fate. A pluripotent stem cell can become any body cell, but could not support the full development of an organism, versus a multipotent cell can become a limited group, and unipotent cells can produce just one cell type. This cellular differentiation hierarchy is key to understanding both development and stem cell therapies. This is showing kind of our stem cell potential, and it follows a well-defined path. Our totipotent cell exists only in the early embryo. Pluripotent cells arise in the blastocyst and can generate all 3 germ layers, which we'll learn shortly. Multipotent cells, as seen here, are tissue-specific, such as hematopoietic stem cells. Finally, unipotent cells, like muscle satellite cells, for instance, are restricted to a specific single lineage. This progression results in increasing cellular specialization and decreasing cellular flexibility. Cell fate is not a randomly determined path, though. Rather, signaling factors in the embryonic environment will guide stem cells down specific pathways. For example, erythropoietin stimulates stem cells to become red blood cells, versus granulocyte colony stimulating factor directs differentiation into neutrophils and other granulocytes. These cues, these chemical cellular cues, ensure proper tissue formation and are also used therapeutically in conditions like either anemia or neutropenia to correct a deficiency. Take a moment to think now. When might the body increase production of erythropoietin, or GCSF, and think about certain physiologic stressors, hypoxia, infection, chemotherapy. These are not just abstract pathways, they are clinically relevant, especially in studies like hematology and oncology. Proteins are the true effectors of cellular identity. Every aspect of cell function, from structure to signaling to metabolic activity, is protein-driven. This includes enzymes, receptors, transcription factors, and structural components. Protein synthesis reflects which genes are active and helps determine the cell's role within the organism. Importantly, differentiated cells activate a subset of their genome. In fact, only about 10 to 20% of genes are active in any typical cell. This selective expression defines the cell's function. The architecture of chromatin, how tightly DNA is packaged, along with the presence of specific transcription factors, will dictate which genes can be accessed and transcribed. So let's consider this. Think about the following cellular types. You've learned most of them throughout the year: fibroblasts, keratinocytes, goblet cells, etc. What unique proteins might each produce? Think of structural proteins like collagen and actin, secretory proteins like mucin, or even signaling molecules like neurotransmitters. These are the proteins that define cell identity and function. When we come back, we will look at epigenetics, or changes that can occur to genetic material to affect its ability and expression.